Our next panelist is Dr. Eric Daniels, with over 20 years of operating experience in the biotechnology and prior to serving as Kiora Pharmaceuticals Chief Development Officer. He served as CEO of OcuRx PT Ltd. And is co-founder and director of Bayon Therapeutics, Inc., both focused on the development of next-generation ophthalmologic therapeutics. Dr. Daniels is also the co-founder and director of Optogen, Inc., a specialty ophthalmic biotechnology company. Dr. Daniels holds an MBA from the Anderson School of Management at UCLA, where he also received his MD from UCLA School of Medicine. And Dr. Daniels received his BS in molecular biology from the University of California, Berkeley.
Well, good morning, everyone. I'm Eric Daniels, Chief Development Officer at Kiora Pharmaceuticals. It's really, truly a privilege to be here amongst members of this community, leaders, fellow researchers. My goal is really to have you leave here with a high-level understanding of what we try to do here at Kiora and where we sort of fit into the broader ecosystem of research that's going on in our space. So I'll get through the housekeeping side of things. We are a small, publicly listed pharmaceutical company based in San Diego, but we have a footprint in Australia as well as, of all places, Vienna, Austria. As a publicly listed company, I'm obligated to show you our disclaimers, but you can look on the SEC website and probably see what I ate for breakfast this morning. So, in a nutshell, at a high level, we are we have two assets in development. One of them is 301, which is what I'm here to discuss today with you. And as others have spoken about, I think we've all determined why we go about doing what we do and the need to be able to develop innovative therapeutics in this space. And just a quick snapshot at our timeline. What you'll find, or rather our pipeline, what you'll find with us is most of our work to date has been in RP, and it really sort of sets the table for the work in choroideremia, as far as what we've been able to focus on and how it is applicable. So that is definitely one of the things I'd like you to leave with, is why our work in RP would be applicable, and to understand some of the sometimes subtle, sometimes not so subtle differences in our approach to how we think about things. So let's get into it. Dr. Mack gave us, as did Dr. Agarwal, a description of the retina. So, I think if I narrow down to what we do, I honestly think of the retina as sort of a layered cake, and at the top of that, there are candles, if you will, and the last layer of that cake are cells that have been mentioned, which are retinal ganglion cells. These are cells that are known to survive. So even though that you have degenerated photoreceptors, those retinal ganglion cells have been shown to be persistent for decades, and it's their tails, if you will, their candle is the one that extends their that information, that electrical information, to the brain, and that's what we perceive as vision. So Dr. Agarwal did a great job of explaining optogenetics. We're really optogenetics without the genetics, and I hope that's clear by the time I'm done with this. So what we do is we actually target the retinal ganglion cells. We try to make the retinal ganglion cells, which normally receive their signal from photoreceptors through other sort of intermediate cells or intermediary cells, we target directly the retinal ganglion cells. And what we add is a small molecule, what's called a molecular photoswitch. And these are small molecules that are fascinating. They have the ability to change shape in the presence or absence of light. So they change their shape or their conformation in the absence or presence of light. And they interact with these ion channels that sit in your retinal ganglion cells. And they get access to that retinal ganglion cell only if they're sitting what is called downstream from degenerated retina and degenerated photoreceptors, to be specific. So if you look at normal retina, there is no ability for these photo switches to get inside retinal ganglion cells. But if it sits downstream of degenerated rods and cones, you get this upregulation, this change in a retinal ganglion cell. These certain pores open up and allow these photo switches to go in. And once they're in, they sit on both sides of this membrane. And in the presence of light, they change shape, and that causes current to go into the retinal ganglion cell. And current going into the retinal ganglion cell is the cell's way of being active and sending a signal to the brain. So the idea is we're sending, if you will, non-native code or new electrical signals from cells that normally play a role, but otherwise, in the presence of degenerated photoreceptors, wouldn't have that activation. So I'm not going to show you the decade of preclinical work, but it does rest on the shoulders of folks at UC Berkeley. It rests on the folks like Russ Van Gelder at the University of Washington. Lots of preclinical studies and safety studies, but it reaches a point where you can only ask so much of a mouse, whether they perceive light or not, or whether they can carry out a test. So we ultimately were able to take this into clinic and do a first-in-man study, which we've already presented data on several the last couple 18 months or so. And we started in a group of patients with retinitis pigmentosa, in particular very late-stage retinitis pigmentosa, folks who either had no light perception or were down to, at least clinically speaking, hand motion or counting fingers. We of course quantify that on some of the same on the similar scales that were presented earlier. And this was a first-in-man open-label study where we're really looking at different doses of the drug applied. Now, a couple of key differences. So we're talking about a small molecule here. The majority of drugs on this planet that most people take are small molecules. It does mean that we do repeat injections, and like Dr. Agarwal's approach at Ray, we also use a standard intravitreal injection, so not a subretinal injection. So we also too don't take patients to the operating room, or our clinicians don't. They just do it in a clinic like they would inject for other millions of other injections that happen all the time. But it also means that we look to do a repeat dose as a small molecule. So every 4 to 6 weeks, we would repeat this. The other major difference that I want to highlight, and this is also important in understanding your entire ecosystem and what options might eventually be available to you, is that we are not a disease modifying approach. We are a vision restoring approach. So we are not trying to slow disease. We'd love to, but we don't. That's not our approach. We're simply trying to recapture what has been lost already by recapitulating the visual system with, in this case, this small molecule approach. So we followed these patients over a course of 30 days in each eye inject. And our first, of course, is obligation, if you will, is to demonstrate safety. So we want to make sure that the drug is appropriate and we're not causing any issues. This is just a table showing that and highlighting that there really aren't any safety signals. We look extensively and are confident in the safety profile of the drug, and the table and the data supports that. I'm going to just kind of go through a handful of efficacy endpoints, meaning that, well, we got to ask people, well, what is this improving? Are you experiencing anything different when you look at the actual efficacy endpoint? So one of the first things we do is a kinetic visual field, or to try to look at their peripheral vision. And you can see over the course of time, over that 30-day period, you see an increase in their peripheral vision, and then it starts to decrease, which again is consistent with the kinetics of the drug. It is a small molecule. It goes into your eye and eventually gets eliminated by your body. So we expect the effect to go down. And then on the right is just another way of demonstrating that change over time relative to baseline. So that's an improvement in in these participants' kinetic visual field or peripheral vision. We also use a particular method of measuring their visual acuity. So all of the patients in our study to date and moving forward, and I'll talk about our next study, are so-called off-chart patients. So these are folks that we can't use traditional approaches to measuring their visual acuity, and so we use other accepted methods, and there are a handful that are out there, and we just use one called the BRVT, and we're looking for reductions in the score, which is the same way of saying their visual acuity is increasing. And we did in fact see that in patients that already had better vision, so patients who came into the study with better visual acuity. We also just did simple light perception. We did have subjects in the study who had a decade plus of no light perception. So we're asking very simple questions. Do you perceive any light? Forget projection or directionality of that light, but just simply, do you perceive it? And that was an interesting result in that for those patients with the most severe form of visual loss, or NLP patients, we did in fact see an improvement, and we measure that in what's called an odds ratio, which is just the simplest way to put it is the strength of association between two things. So you like to see odds ratios greater than one. If you have this input, you expect to see, not expect, or there's a correlation or a strength of association with an output. In this case, the input is they're on drug. The output is, can they see light or can they perceive light any better? But again, we see it declining over time at that peaking in around that 2 to 3 week period and then declining, which again is something consistent with having a small molecule approach. We talked, or Dr. Argowal talked about these functional tests. They really do play an important role in our world because they have traditionally served as the means by which regulatory agencies around the world approve these types of therapeutic approaches, which is, can people carry out tasks that they otherwise couldn't do before on drug, in particular at lower light levels. So we asked people to go through a series of different tests and functional tests. And this was our first kind of pass at this. We've subsequently refined a handful of these because some of them looked were reliable, others weren't. Others were incredibly fatiguing over time, and they just simply weren't something that would be repeatable and sustainable for future studies. And so, but we see a variety of different responses across those different tests, and again, it gives us comfort that we are seeing some improvement in this type of assessment. And then we simply ask, using standardized questionnaires, whether their visual lives, if you will, are improving over time, over that course of 30 days. And that is also something that we saw. So one of the probably the more interesting things that we elected to do is, I mentioned to you, we're trying to activate the top layer of the cake and the candles. And those candles travel to the brain. So we thought, well, wouldn't it be interesting to just actually look and what is the brain itself lighting up? So we can do that today with something called functional MRI, where we can show patients images and we can measure whether their brain is active or increasing over that period of time. And you can see in this sort of panel of different subjects who participated that you see increasing activity in the part of the brain responsible for visual perception, that back part, that occipital cortex. And you can see that over time, that it increases, sort of peaks at day 14, that 2-week part, or that 2-week time point, and then starts to decrease at the 30-day window. Again, all kind of consistent with the kinetics or the timing that we know about these small molecules. And if you quantify all of those things, they do appear to have more signal on than they do off. So it was nice to sort of see that correlation. So when we look at the study, our goal from this first-in-man study is not to say at all, hey, we've got a treatment out there. What we have is, we are looking for some signal. We're looking for something to tell us in a compelling way that doesn't appear to be just from random chance, which is very hard to do. And we're not suggesting that we did that, because we were dealing with small sample sizes, we're dealing with non-controlled study, but we're looking for some sort of non-random signal that says this is worth continuing to pursue, and that we've seen enough, and that naturally that it's safe. So that's where we stood about 12 or so months ago. And you can visit our website, and these are some of the testimonials from patients, which are obviously extremely powerful, and they're great, but they have to be supported naturally as well by the objective data. And for us, it's really the compilation of all the different types of data, no matter how we looked at it, whether it was visual field, whether it was the functional test, whether it was MRI. They all seem to be pointing in a direction of positivity or directionality. So that leads us to our next study, which is called Abacus 2, which we're about 60 days away from dosing our first patient. There are going to be 3 big differences in that study relative to what we did in our first study. The first is that we do have a control, so there will be subjects or participants or patients that do get a placebo, if you will, or in this case, a vehicle control. Now, all of those patients will eventually have the opportunity to participate and have active drug at the end of the study, or at the end of the first part of the study. So everyone, if they wish to do that. The second part is that I mentioned in our first study, we did a single injection. We'll be doing repeat injections. So every 6 weeks, we will be giving folks injections, and we'll do that 3 times. And in the first study, we injected or treated only one eye, and we will this time be using both eyes. So we'll be doing this bilaterally. So those are the 3 big differences. We do our work in Abacus 2 currently all within 5 sites within Australia. I mentioned we have a footprint in an office and we do a lot of clinical work there. We can get into that, or you can ask me later why that's the case. But I can certainly give you, and if you visit our website, you can be taken to a host of resources that can give you the inclusion and exclusion criteria. Again, these are folks with RP, not choroideremia, but it will serve as the foundation for moving into additional inherited retinal diseases. This is also where I would pause and say that we are like Dr. Argowal explained, we are a mutation agnostic approach. So no matter what the underlying mutation is, because we aren't trying to recreate or solve for a particular mutation. It's not that we don't care. It just is we're agnostic. So we're bypassing that machinery, which is also, I think, we like to think, an attractive approach. We are changing up some of the functional tests that we use based on feedback from regulatory agencies. We're currently refining that. This is an area where we have worked with the CRF, and thank you to them for their ongoing support in the form of a grant, and thank all of you. So we are undergoing that retest validation of some of our current tests that is ongoing now and wrapping up, and the data does look quite good, so I'm happy with that. And I also want to thank, I don't know if I had it up here, but we have been fortunate enough to have partners in the field. So we have a relationship with, we are a very small pharmaceutical company. We have a relationship with 2 sort of mid-sized, bigger pharmaceutical companies who are very keen on what we're doing. One is a French group called TEA, and the other one is a Japanese group called Senju. We have partnerships for this drug with both of those pharmaceutical companies, and that gives us the ability and the financial wherewithal to make sure that this important work gets done. So there is effectively, a very good probability that one day I'll be on stage, hopefully in the same group, telling you about data from there and how we're moving next to choroideremia. So thank you very much for your ongoing support, for your time, and appreciate everything.
Thank you very much, Eric. We're a little behind, not because of our speakers. There's no question about that.
I can take a hint, Neil.
Sorry, Eric.